Every disease has a protein signature. Cancer, HIV, Alzheimer's, all leave behind specific proteins that tell scientists exactly what is happening inside your cells. But how do scientists detect these specific proteins from millions of others? That's exactly what Western Blot does. Let's understand how it works. Western Blot is a laboratory technique used to detect specific proteins in a sample. It tells you, is this protein present? How much of it is there? What is its molecular weight? Western Blot is used in disease diagnosis, research laboratories, quality control in biotech, HIV confirmation testing. Think of it as a protein fingerprinting technique. Just like fingerprints identify people, Western Blot identifies specific proteins from a complex mixture. It was developed in 1979 and remains one of the most important techniques in molecular biology today. First, the protein sample is prepared from cells or tissue. The cells are broken open using a process called cell lysis. This releases all proteins from inside the cell. A protein quantification assay like BCA or Bradford is performed to measure protein concentration. Equal amounts of protein are loaded for accurate comparison. The protein sample is mixed with a loading buffer containing SDS. SDS is sodium dodecyl sulfate. It denatures proteins and gives them a uniform negative charge. The proteins are then loaded onto a polyacrylamide gel. When electric current is applied, proteins migrate through the gel based on their size. Smaller proteins move faster, larger proteins move slower. This separates proteins by their molecular weight. A protein ladder is always included to identify protein sizes. After electrophoresis, the separated proteins are transferred from the gel onto a membrane, usually a PVDF or nitrocellulose membrane. This transfer is done using an electric current in a transfer buffer. The membrane is much easier to work with than the gel for antibody detection. The membrane is blocked using 5% milk or BSA solution. Why blocking? The membrane has many non-specific binding sites. Blocking fills these sites to prevent false positive results. This step is critical for clean and accurate results. Blocking is usually done for 1 hour at room temperature. The membrane is incubated with a primary antibody overnight. At 4 degrees Celsius, the primary antibody is specific to your protein of interest. It binds only to that specific protein on the membrane. This is the detection step. After washing off unbound primary antibody, a secondary antibody is added. The secondary antibody, right-pointing arrow, binds to the primary antibody. Right-pointing arrow is tagged with an enzyme, usually HRP, horseradish peroxidase. Right-pointing arrow amplifies the signal. This step makes the protein visible for detection. Finally, a substrate is added that reacts with HRP enzyme. This produces a chemiluminescent signal that exposes the X-ray film or is captured by an imaging system. The result is a band on the membrane, at the exact molecular weight of your protein of interest. Reading Western Blot results is straightforward. A band at the expected molecular weight means your protein is present. Checkmark. No band means protein is absent. Cross mark. Multiple bands may indicate right-pointing arrow non-specific binding, right-pointing arrow protein degradation, right-pointing arrow post-translational modifications. Always compare with positive control, negative control, loading control like beta-actin. This ensures your results are accurate and reliable. Western Blot is one of the most powerful protein detection techniques in molecular biology. Master this technique and you will have a significant advantage in any research laboratory. If you found this helpful, subscribe to Molecular Bytes, where we simplify molecular biology 1 byte at a time. Next video we will cover how to troubleshoot common Western Blot problems. See you in next video.